So there are a number of excellent reasons to go after very high-quality genome assemblies. And myself and Manyuan Long at University of Chicago and Rod Wing put heads together and came up with a number of questions for which you really couldn't do any progress without much better assemblies than we had. So Manyuan and I had been involved in the 12 genomes project with Drosophila, where 12 different species had been assembled by Sanger sequencing methods, and we thought, well, let's update that by looking at the latest. And so the basic idea was to apply, again, comparative genomic methods, but with very high-quality PacBio assemblies. So the three primary questions that we came up with that were convincing enough to get us an NIH grant for it were first this one, which is Manyuan Long's thing for over 20 years now, looking at de novo genes, identifying genes that aren't present in the ancestral species, that are novel genes that require very, very good assemblies, of course, to have confidence in those. Heterochromatic regions of the genome. So we haven't heard anything about this topic yet in this session. And it's the area of the genome that is the most difficult, highly repetitive, often pericentromeric, peritelomeric regions of the genome that are of great functional importance. You need telomeres, they need to remain intact. You need intact centromeres to have chromosomes segregate properly. But these regions of the genome are really a bear to work with. So can we make progress with long-read sequencing there? And finally, transposable elements. We can do a pretty good job with transposable elements with other kinds of assemblies, but one of these platinum quality genomes really lets you have high confidence that you've identified all the transposable elements. And with those, we can also identify the host defense against those elements, namely through the production of piRNAs. These are generated by piRNA clusters that are transcribed through very, very difficult regions of the genome to assemble. I'm only going to focus on the middle one today. So thanks to Manyuan generating the inbred strains, this was all based on collections of flies from inbred strains. Many of them were from the previously sequenced lines. Those were shared with Rod Wing's group at University of Arizona, and this just shows a lot of the raw data from these. We were getting between 100 130x coverage of these flies, typical read length averages of about 10 to 14 or so kilobase pairs. It's somewhat harder to make really long molecule genomic DNA from flies. The assemblies, however, flew together very nicely, often getting contig N50s of 15 to 20 or so megabase pairs in length, so almost whole chromosome arms assembling into single contigs, with longest contigs on that order of 30 or so kilobase pairs, sorry. So many of these species do have a lot of repetitive DNA. This is showing human, axolotl, and maize, and you see there's quite an abundance of transposable elements in all three of those genomes. Human genomes about half transposable elements, but not so much of these simple repeats. All genomes do have some degree of so-called alpha satellite or very simple repeats around centromeres, but not very much in those vertebrates, or in maize. On the other hand, Drosophila virilis is 40% of the genome is simple repeats, so quite a dramatic expansion. What the heck is going on in those genomes? Why do they have them? What's going on? Drosophila melanogaster, on the other hand, is much less. So even from one fly to another, considerable variation in the amount of the genome that's taken up by this junk. Okay, so the virilis genome actually had been known since the 1970s, using cesium chloride density centrifugation. You get a sharp band of these materials, hence the name satellite repeats. And those could be sequenced and identified that they were often very, very long runs of 7-mers, for instance, that were repeating. And the genome size of Drosophila virilis is a real outlier. It's like 400 megabase pairs compared to roughly half that for many other fly genomes. And so the implication was that a substantial fraction of that expansion of the genome of some of these species is simply these simple satellites taking off. And so from the sequence, we knew that it was primarily 3 or 4 of these 7-mers that had gone wildly astray in Drosophila virilis and expanded. So some of the questions: is it really the case can we show this much more rigorously now with these long-read sequencing methods, that almost half of the genome is 7-mers. Can these sorts of methods more accurately describe these repeats in terms of, are they in large contiguous blocks? Are they spread all over the genome? What's their organization in the genome? What's the variability from one line to another? Are they rapidly evolving? All those sorts of questions. Why and how do these do species that have so much acquire so many of these repeats? And again, this is just showing the phylogeny of the species that we looked at, and it's an expanse over more than 40 million years of evolution. So these repeats are often assembled very nicely from long reads, not the alpha satellites so much, but more the transposable elements and more complex kinds of repeats rather than these simple repeats. And this is one of the big take-home sort of wow figures from the our experience anyway in doing these assemblies. Drosophila ananasi chromosome 3 assembled virtually telomere to telomere. That is clear through the centromere. We have a single contig. This is just showing that there was a region of the genome. The top is showing the simple sequence repeats, and you can see that it drops down in a region in the center of the chromosome around the centromere. Essentially right at the centromere, there is, again, a spike of these simple sequence repeats, and then it drops back down. And that corresponds to a dip in the transposable elements. So we have this sort of segregation between transposable elements versus simple sequence repeats around the centromere of Drosophila ananasi. Now, Barbara Malone and others have been doing some beautiful work actually identifying the location of the kinetochore by doing CenP, the centromeric histone chromatin immunoprecipitation, to identify exactly what is the functional element of the centromere. And it often turns out to be these small sort of islands in the middle of those regions of the chromosome. It's also very easy to identify these somewhat more complex, longer repeats, and we've discovered a number of longer repeat families within the genomes that we've been assembling. So mega base pair lengths of these runs of 32 mers, 36 mers, and so forth. The sad news, however, is these very simple repeats, the things like the 7 mers, do not assemble even with PacBio. And so here we see the assembly from the original Sanger sequence had almost 7 or so megabase pairs of that AAA-CTAC 7-mer, whereas the PacBio and the nanopore assembly from Dan Miller's group have only about 29 kilobase pairs of that sequence. So there's a huge dropout from the assembly. They just are sort of discarded by the assemblers. Nevertheless, we don't need to rely on the assembly to make some sense of them. We've come up with methods of going just back to the raw reads. And we have a very beautiful collection of raw reads from the PacBio sequences that are very long. And for instance, here's a 50 kb read that's nothing but that 7 base pair repeat. And if the animation worked, you'd see this scroll, and you can actually see the sort of patterns go as a moire pattern across the screen. So it's really quite dramatic seeing these repeats. And we can tally those up now and get a picture of what are the distribution of these numbers of repeats. And so comparing across closely related species like Virilis novomexicana and Americana, which isn't on this picture, but what you can see is that relatively recent ancestry may have an absence of a particular repeat, and then they take off. So Virilis, compared to its close relative, has more than double the abundance of that particular 7-mer. And so it's obviously rapidly evolving. We've done, separate from this study, mutation accumulation experiments, which show that these things are ticking along at about the rate that microsatellites are. That is, in other words, they're changing in copy number about 10 to the minus 4 copies per copy per generation. And so even two individuals from the same line, given that you have millions of copies, will have hundreds of copy number differences between them. And so this makes it a challenge to work with multiple individuals, even in an inbred line. And so Julian Flynn, who's the graduate student who produced many of these slides, and this is a part of her PhD thesis now, got very good at doing fluorescence in situ hybridization. And this is the best way to go from the step of getting the reads, where are they, to looking at actual locations in the genome. These genomes, Virilis and novomexicana, are all telomeric, a little bit unusual for a Drosophila genome. And this is just showing in that, I can't point very well, the cell right above that text that shows the sequences. You can see the identification of these repeats, and the green one is that very abundant telomere. You can see it's pericentromeric on all the chromosomes in Virilis, whereas in Nova Mexicana, it's only one of the chromosome pairs has that pericentromeric location. So very rapidly, it gets onto another chromosome and expands. And that's the process that we're trying to explore by looking at multiple lines. And towards that goal, we've decided that it would be really nice to sequence multiple other either individuals or lines, and we sort of posed the question, would it be possible to do PacBio sequencing with single individuals? And the PacBio people very happily jumped to this challenge and even supplied me with a couple of slides. These were only delivered within the last week, and so we've only barely started to look at them. But as Justin mentioned, this Express Prep 2 is able to work with very small amounts of DNA, and even more quickly than previous methods. And so what was done was to generate libraries from single flies, 5 single Drosophila erecta flies. A single fly will get you about a microgram of DNA. These were started with about 150 nanograms. And in one case, there was a 30x covered genome sequence produced with just 29 nanograms of input DNA. And this is just showing the using cleanup procedures that are actually losing even more of that DNA, but it gains you in the quality of the sequence input to the library preparation. You get very consistent, reasonably high quality libraries from this approach. So the pre-assembly metrics, again, are showing this sort of reasonable length distribution on the order of 10 to 14 or so. KB is an average read length. And then the assemblies come down to pretty much the whole genome. The whole genome of Drosophila erecta is estimated at 145 megabase pairs. These assemblies are coming in at 139, 138. So the one thing about erecta is it has way less of this simple sequence repeats and assembled beautifully by this method. Okay, so quick conclusions. These high-quality whole genome de novo assemblies are possible with, in fact, rather routine now with these PacBio methods. These heterochromatic satellite repeats don't assemble, even with these methods. One thing I didn't mention is that, of course, you do get reads that anchor in unique sequence and then go into these repeats. And those are very interesting because you're getting the edge of the arrays of these satellite repeats, and we've been studying those. We can also make a great deal of progress in learning about heterochromatin turnover simply by analysis of these sequences at this read level. And the Virilis group is a particularly interesting group because they're close relatives. They actually readily hybridize, so we can even test for chromosome segregation among individuals that have radically different pericentromeric heterochromatin, which is a key issue of the sort of theories about the evolution of centromeric DNA, that there's maybe a centromeric drive, as Steve Henikoff and Harmeet Malik have posed. And finally, this method to develop whole genome sequences from single individuals is terrifically exciting in terms of the kinds of new questions that we can generate and answer with those data. And so here's Rod and Manyuan and my group, and I'll leave it at that. Thank you.